up next, followed by Melania Figueroa, and finally, Les Szabo will finish it off. And at this point, I'd like to introduce our next speaker. Dr. Cathie Aime. Cathie Aime is a professor of mycology in the Department of Botany and Plant Pathology and director of the Arthur Fungarium and Kriebel Herbarium at Purdue University and a Purdue University faculty scholar. She received her MS and PhD in biology at Virginia Polytechnic Institute and State University under the guidance of Orson Miller, and conducted postdoctoral research at the University of Oxford under Lorna Castleton. Cathie's research combines expeditionary fieldwork and traditional approaches with molecular genetics and multi-omics approaches to understand fungal diversity and evolution. Areas of specialization include tropical basidiomycetes, systematics of early diverging basidiomycete lineages, including smuts and yeasts, evolution of rust fungi, and epidemiology of tropical tree diseases. Cathie is a former officer of the MSA and past managing editor of the journal Mycologia, and is currently vice president of the International Commission on the Taxonomy of Fungi, an executive committee member of the International Mycological Association, and vice president of the Mycological Society of America. Cathie is a fellow of the Mycological Society of America, the American Association for the Advancement of Science, the Explorers Club, and the Linnean Society of London. And with that, please go ahead, Cathie, with your presentation.
Thank you very much. Should I share? Let's see. Can you see it all right?
Yes, I can see the slides.
Okay, great. Listen, I do want to thank you very much for inviting me to this. Obviously, I don't work in any particular pathosystem, so I'm going to be talking about something a little different. But I do want to acknowledge Les and the influence his thinking about rust fungi has had on my career. I didn't start working with rusts until I joined the USDA in 2003, after I got back from Oxford. And Les was probably the first uridanologist who came to visit me at the USDA when I started working with rust. And I still remember looking at some work he had brought with him, some slides of things you were doing, Les, with allium rusts, and especially the interesting results you were having about the influence of the ecial host on some of those pathosystems. And it really got me thinking a lot about the evolution of this group as a whole. And so that's what I want to talk about really today. And if I can get my slides to work. There we go. So I'm not going to spend any time on this. Everybody knows what rust fungi are and why they're important in agriculture. But the things about rust that really intrigued me and why I became interested in them as a biologist is because they combine a lot of features that are really unique or rare in fungi otherwise as a whole. Many interesting characteristics. They're of course all obligate biotrophs, and that's a rare strategy in fungi. There aren't too many fungal groups that have radiated or specialized as absolute obligate biotrophs. Certainly the rust fungi possess some of the largest genomes that we know of in fungi, but the real interesting factors of their life cycle are the fact that they have this alternation of generations. So they produce separate gametothalli and sporothalli. And these gametothalli and sporothalli, of course, are produced on two different unrelated host plants for most of these rusts. And this, again, is really unique to fungi. There are a few aquatic things that produce an alternation of generations, but when you combine that with the heteroisism, it's really, as far as a successful strategy, confined to just the rust fungi. And then in addition to this alternation of hosts and the alternation of generations, we have the production of up to 5 different morphs in any rust life cycle, which again is a very unique strategy that's limited to the rust fungi in this case. And because of this complexity of these life cycles, of course, the life cycle data are missing for many. This isn't a surprise to anybody listening to this, but it is important to keep in mind that when we try to dissect out evolutionary trajectories for the rust and their hosts, it's a lot of the inferences we draw are based on the fact or limited to the fact that we are still missing life cycle data for many of these species. So very, very quickly, because I started out as a systematist before working in molecular genetics, the rust fungi, just to be clear, when we talk about these as a group, we're talking about a single order of fungi, and this is the order called the Puccineales, and it's a tremendously successful order of fungi. There are 18 families as we circumscribe them to date, with about 170 or so accepted genera and about 8,000 accepted species. And when you take that all together, that means that the rust fungi themselves, this one order, the Puccineales, it is the second largest order in the fungi. The only larger order really is the Garicales, which contains our mushroom fungi, which are the ones that are really easy to see and describe. So this is an incredibly successful group in terms of species diversification, with so many species in the one order. And basically the rest make up about 8% of our described fungi, or a quarter of our basidiomycetes. And they are the largest group of plant pathogens, the largest natural group of plant pathogens. And this sort of brings us to the conundrum about studying rust fungi, at least as it is in the literature, and that's the fact that we have an incredibly successful group, again, in terms of species diversification, but one in which these incredibly complex life cycles have evolved, and it's difficult for people to reconcile the evolution of these complex life cycles with extreme success. John Bonner, who was worked at Yale, worked on slime molds, the cellular slime molds. And to Bonner, understanding an organism's life cycle was really key to understanding evolution of organisms. We tend to think about organisms almost statically, at least in mycology, because you're typically collecting these fruiting bodies in a very specific place in time, but that doesn't tell you anything about the life cycle. And to understand the evolution, of course, we need to think of these things as complex wholes. Now, complex life cycles, one interesting thing about these, so our modern day definition is that a complex life cycle, of course, pertains to species that must infect more than one host species to complete their life cycle. So that would be anything, for instance, that's heteroecious. There is another modern definition that distills this down to basically any organism that undergoes some sort of abrupt autogenic change during the life cycle, which again is also applicable to rust, because these 5 different morphs, each of these is a very abrupt onogenic change, if you want to think of that, or punctuation during the completion of the life cycle. Now, what's interesting about complex life cycles is, of course, these things have evolved several times independently throughout the tree of life, or at least throughout the eukaryotic tree of life. But most of the work that's been done understanding complex life cycles is work that's been done with human pathogens for the most part, things like Amphicomplexa, which give us malaria, or the parasitic helminths. And in most of these cases, we are talking about single-celled organisms. And again, our understanding about complex life cycles really just derives completely from these animal-based systems. What's interesting about the rust is that they have a very specific type of complex life cycle that we call the haplodiplonic life cycle. And that means that these are actually multicellular in both the diploid, that's actually dikaryotic, of course, for Rus, but in the two nuclei and the haploid stage, they are multicellular, again, in contrast to most of these animal pathogens. So the life cycle, this is the complex life cycle, or the blueprint for rust. And this, of course, a Melampsera species we have to illustrate it here. I know you all are familiar, so I won't spend too much time on this, but I do want to point out a couple of features that make these pathogens really distinctive. And again, parts of the life cycle that we don't find in other fungi. For instance, the basidiospores in other fungal systems, the basidiospores or ascospores are the haploids, and those are the basis for fertilization, right? So you have these two haploids get together to fertilize. But of course, that's not the case with the rust. They have this amplification of the haploid step in these spermagonia. So if, for instance, you have a mutation, as we just saw in the last talk, in one of your effectors that happens to one of these haploids, it's not just a one-off chance that it's going to meet a compatible mate, because that haploid gets amplified. And these spermatia, of course, a single spermagonia can produce thousands of identical spermatia, which are all capable of mating with other compatible spermatia. And of course, we know that insects and wind are capable of carrying these spermatia to other plants. So any beneficial mutation that happens during meiosis here can in effect be widespread and cause multiple matings through this amplification step here in the spermagonial stage. And then once fertilization, of course, takes place, we have this other unique stage here. We do have, of course, conidial stages. Something analogous to this would be a conidial stage in your pathogenic ascomycetes. But in rust, this stage, this uridinospore stage, which is our clonal repetitive stage, can continue indefinitely. All it takes is a good host and good environmental conditions. And we've seen that this happens in the spread of a lot of our emergent rust diseases. If the uridinospore stage is on a host of economic importance, such as Ostropoccinia acidii, or Phacops pachyrizae or Hemilia vastatrix, then it is capable of continuing in this clonal asexual phase indefinitely, given the right host and environmental conditions. So these two amplification stages are very important for understanding how rusts evolve and how they are so easily overcome host resistance in many cases. Now that's this typical type of life cycle here is one that we call the heteromacro cycle, because we are talking about two separate hosts that are host to the gametothallus and the sporothallus, and the 5 different morphs or spore stages that make up the macrocyclic life cycle. But we do know, of course, that rusts, in addition to that heteromacro cycle, do have other innovations in their life cycles. One example of this would be the things that we call demicyclic, and these rusts, Gymnosporangium species would be a good example, actually eliminate this uridinospore stage. So they don't have that clonal dikaryotic repetitive stage, although they certainly have the haploid repetitive stage. Demicyclic rust can be either hetero, so they produce this sporothallus and gametothallus on separate hosts, or they can be audacious and produce them on the same host. And then we have things called microcyclic rusts, which have actually eliminated the eschal and the uridinal stage. So they basically eliminated the clonal dikaryotic stages, although they can still produce spermagonia, so they can still produce those clonal haploid stages. And then we even have things called endocyclic, which are rusts that have basically morphological sori that look like Escherichia spores, but they are capable of reinfecting the same host from which they came from. And not only that, they are capable of serving as the proboscidium where meiosis takes place and producing basidiospores. So these endocyclic are not the same as the microcyclic rust, but it's a specialized case where you have meiosis occurring in an Escherichia, what was once morphologically an Escherichia at any rate. And so one of the questions, actually, when I began working on rust fungi, we had a lot of questions about how these things evolved, and there were a lot of hypotheses to test. But of course, if you want to understand the evolution of a group, especially a successful group like this, you need to know what the sister group is, and that was not known back when I began working on these. So that was one of the first things we set out to do. And you're just looking at the entire subphylum here. And all the different classes, and here are the rust fungi. And we discovered, of course, that the sister group to the rust is this little known order called the Platyglobioales. And there's two things about this that I want to point out. One is that in the Platyglobioales, we see they produce a fertile hymenium, so it's unlike rust in that respect. But this hymenium consists of these teleospore-like proboscidea that germinate to form these regularly transversely septate basidia. So very rust-like in that respect. Of course, the formation of this hymenophore on an actual, in this case, clavarioid type fruiting body is not rust-like, but certainly the production of these proboscidea, teleospore-like cells are. And this is a fern pathogen called Eocrinartium, and it turns out that the Platyglobioales have many fern pathogens, well, many, in this group, that means about 10 or so, but they have fern pathogens that all have this type of life cycle. Very, very simple. These basidiospores do not undergo any amplification. They simply mate and reproduce the fruiting body and these proboscidea. So in that respect, it's not rust-like. So that gave us a hint, though, that at least the ancestor to the rust probably had some sort of proboscidea or teleospore. But did they have these other life cycle stages? And that's something that was not known. You know, there were hypotheses that rust evolved from very simple life cycles, and each spore stage was kind of added on during the course of evolution, or that perhaps the reverse might be true. And so this, again, is early work where we set out to sort of resolve the rust tree of life, which we were not able to successfully do at this time, but we could resolve the base of the tree of life for the rust. And what you're looking at here is basically the base of the rust tree of life with the different characteristics as far as these different morphs. 5 different morphs are concerned. So we have our outgroup that forms these proboscidea, teleospore-like proboscidea. And then our earliest diverging extant rust is something called Roger Petersonia torrhea. I just got back from California yesterday, where again, I was searching for the alternate host of this thing for 2 weeks, and we came up short again. So as far as we know, this particular rust only produces a spermagonian Escherichia. We still do not know if it has an alternate host or what that might be. Really, instinct is telling me this alternate host is extinct, because the host of Roger Petersonia torrhea is this very relictual plant that's only still extant in a couple of populations in Northern California. Its range has been greatly reduced, and it looks like it may no longer have a teleal host, and it just persists as this gametothallus. And then our next group of rusts are things like Hemilia. Hemilia vastatrix is the coffee rust fungus, if you're familiar with that. But what's interesting about the Hemilia is we have about 70 species in this group, and they are all only known from the uridinyl and teleal stages. We don't know any gametothallus for any of these. They produce functional basidiospores, but we have no idea what those basidiospores infect. And then the sister to the Hemilias is actually this small group called the Micronegariaceae, and this includes things like Micronegaria, which are fully macrocyclic and heteroecious and do alternate hosts and produce all 5 spore morphs. So putting it all together, what do we infer from this? Basically, our working hypothesis is that the earliest rust, the ancestor to our rust fungi, did possess these key features of the alternation of generations, the heteroism, probably all 5 spore stages, although it's still unknown whether it produced uridynia, but certainly probably had something equivalent to a telium. And they were obligate biotrophs, having emerged from a lineage of obligate fern or moss pathogens. And so this brings us to, again, that basic paradox of the rust fungus life cycle and the rust fungi themselves, which is, how do you explain such a successful group that has such complex life cycles? It would seem to be counterintuitive that these would be able to speciate so well, given the constraints one can imagine on having to have 2 different hosts to complete the life cycle. And again, most of the work on complex life cycles, as I've mentioned earlier, is done in animals. They have 2 different explanations or hypotheses for this in animal pathogens. And one hypothesis is simply that these onogenic shifts are directed towards maximizing growth. And this makes absolutely no sense in the case of these plant pathogens, because we don't define success here in terms of increased mass or anything. It's just increased capability of producing propagules. And then the other explanation is something called upward incorporation. And the idea here is that if you're a pathogen of a host, and that host itself is preyed on by another organism, then it makes sense for you to actually become the pathogen of the predator of your original host. That's called upward incorporation. That way you don't get eaten too. And again, this makes no sense for rust fungi because plants do not eat other plants. We do know that in plant pathology, we have 2 hypotheses for explaining the evolution of plant pathogens. And both of these may be true in different pathosystems, but one is, of course, host switching. And that is shown here on, this would be an idealized phylogenetic tree of pathogens in their hosts. And if you're a host switcher, then your phylogenetic tree does not pattern that of your host, of course, because you're able to jump across hosts and across phylogenetic lineages. And this requires very little energy as far as initial host specificity, although for this system to work, of course, these different jumps do eventually require some sort of increase of specificity on your host. And then the other explanation we have, of course, is coevolution. This is something like the red queen hypothesis, where you basically have pathogens and hosts evolving together in lockstep. So which explains the pattern for rust fungi. And of course, we have even modern day completely different adherents of both sorts of hypotheses for how rust evolved, whether it's coevolution or host jumps. And so this was a question really that I set out to look at several years ago. And from this early work, this was an early 2006 paper that we didn't fully resolve the rust tree of life, but something interesting that emerged from this work was that if you just looked at the ecial host, or the host on which the gametothallus forms, there is a pattern discernible. These early diverging rusts form their gametothallus on very early diverging gymnosperms. Then we have an entire Pinaceae group, and then a massive angiosperm radiation. And so this gave me an idea of a way we could test whether we were looking at biological specialization in the rust or biogenic radiation. And the way we did this is not as complex as this slide might lead you to believe, but I did this work in conjunction with my postdoc at the time, Andy Wilson here. And what we did is selected exemplar rust from across the rust tree of life. And we wanted to make sure that we had all the different lineages exemplified in our selection and our sampling selection, but also that we picked lineages for which both hosts were known wherever possible. And we simply constructed 3 different trees: our rust tree, and then the hosts for the ECL stage, a separate tree for them, and a separate tree for the hosts for the sporothallus stage. And then with this reconciliation analyses, what you're looking at here is the number of steps involved to reconcile one of these host trees with the rust tree. And no matter which model we used, the results were the same, that it was much, much more difficult to reconcile the sporothallus host with the rust phylogeny than the gametothallus host. The gametothallus host and the rust phylogeny mirror each other very well, is another way to talk about this. So what does it all mean? Well, basically, what we hypothesized in this paper is that, in fact, both of these processes, these biological evolutionary processes, are working on the rust fungi. They're just working on different stages during the development. So we have biological specialization really explaining the patterns that we see between rust fungi and their gametothallus host, versus biogenic radiation explaining the patterns that we see between the sporothallus host, or the sporothallus host, and the rust fungi and the sporothallus. So both of these processes are working. It makes good biological sense. Again, we already talked about these spermagonia, but in order for mating to be successful, these are rather temporally and spatially confined to a single leaf in a single space and time. So you're going to have much more constraint on undergoing host jumps here. This is the perfect sort of condition for biological specialization to really be driving the evolution in that direction. And then again, in the uridinial spore stage, our other amplification stage, of course, these don't have such constraints on them. You can even see it looking at the rust tree of life, that you have a lot of teleal or sporothallus shed, basically driven by these uridinial spores that can shed out on other ecologically co-distributed hosts. While that begins to make sense to us about the evolutionary drivers of speciation in the rust, it doesn't tell us really how these other types of life cycles evolved. And so to really look at that question, we basically tested Transgell's law. Now, you guys all are uridinologists as well, so you're familiar with Transgell's law, which was actually coined by Arthur, but it was based on an idea of Transgell, a German worker in 1904, who believed that these macro-heteroforms were ancestral and that the microforms were derived. And this was due to a whole lot of observation he made in the field, that a lot of the microcyclic species would share a host with a heterospecies, but a different stage. So this is a sporothallus stage here on a ranunculaceae on a microform, but on a heteromacroform, that same ranunculaceae would be host to the gametothallus. And so he thought that this was a simple reduction and that microcyclic species were actually derived from these heteromacro. And so Marcus Scholler and I set out to test this idea. Fittingly in the genus Transgellia, now named for Transgell, but because I'm going to run out of time, to make a long story short, we did find lots of evidence for what Transgell and Arthur would have called correlated species, which is derived microforms being derived from these heteromacroforms and sharing that one host. So this was the model that Marcus and I proposed to try to explain rust evolution and why perhaps there are so many species of these things. And basically you have your heteromacroform, as we've shown in Transgellia, being a pathway for the evolution of new species, new microforms. Whoops. And then of course through time, you also have biological specialization and biogenic radiation through time, creating new species that are heteromacro, and those could also give rise, of course, to new microspecies as well. So ad infinitum, you can see how this opens up some new pathways, primarily in this direction for speciation. What we hypothesized in this paper is that there might also be the reverse might be true, that the sporothallus host, if separated from the gametothallus host, might eventually lead to speciation in this direction. But we couldn't find it down at that granular species level that we were looking with Transgelia. And so to test the idea that perhaps speciation might also be occurring in this direction, we needed a much better better resolved Rust tree of life. And this was done very recently with another former postdoc. This is Alistair McTaggart. And I won't go into, again, all the details of this tree, but basically using type specimens for 80% of the known Rust genera, we were able to fairly well resolve this tree, at least enough to look at that specific question. And for that question, I want to just focus in on this particular suborder here known as the Ravenelliinae. Most of our Ravenelliinae are tropical fungi, so you may or may not be familiar with them. But what's interesting is that this suborder contains 4 families. The 2 earliest diverging of these families, the Ocropseraceae and the Transgeliaceae, contain heteromacroforms, as I just showed you with Transgeliaceae, for example. The next family to emerge in this suborder is the Phycopseraceae. And what's interesting about Phycopseraceae is that these are only known to date, even though there's well over 100 species, close to 200 species in this family, they are only known from the sporothallus. We do not know gametothallus for any of these. They produce viable basidiospores, but those basidiospores, at least in the species that have been tested, do not reinfect the same host from which they were produced, so they are not audacious. And then finally, we have the Ravenelliaceae, the last family to emerge in the Ravenelliinae. And the Ravenelliaceae are all audacious and macrocyclic, so they produce gametothalli and sporothalli, but they produce these all on the same host. So again, just to summarize real quickly, what do we think this all means? And basically, we think this is evidence for speciation occurring on this sporothallus host as well. So for instance, let's say you have a heteroecious macrocyclic rust and some perturbation or local extinction causes the separation of the gametothallus and the sporothallus. So what happens? Well, we've seen in Transgelia that these can speciate to become microcyclic forms. And what we think is happening then on the sporothallus host is that these can persist, as we've seen in Phycopseraceae, for a long period of time as just uridinospores and teleospores. But once, perhaps in a blue moon, these do produce basidiospores, and that very, very rare chance that some of these basidiospores become capable through mutation of infecting that sporothallus host, they quickly compensate by reforming the other so-called lost morphs, the Spermagonia aecia and uridinospores. And you can really see this. I realize this is difficult perhaps to see on this screen, but in the Ravenelliaceae, if you look at the tree produced by standard phylogenetics, for instance, you know that phylogenetics forces a sort of dichotomous evolutionary pattern, even if that may not be the correct representation of evolution on any given data set. If we actually produce a network analysis or conduct a network analysis of those same data, we see actually that the Ravenelliaceae, as you can see here, is a massive radiation of species. We don't see this pattern in any other parts of the rust tree. And again, what we think this means is that eventually one of these basidiospores became able to reinfect the same host on which it was produced, and that just supercharged this massive radiation in this group. Do we see massive radiations in microcyclic or endocyclic rust? And the answer to that right now is no. We only see it from these sporothallus hosts. not yet from the gametophyllous hosts. And in fact, these microcyclic and endocyclic species always seem to be terminal points. So that seems to be not a leading cause of radiation. And therefore, we think that really the drivers of radiation are probably these uridinospores. Once those get reconvened, we see massive bursts of radiation. And so our pattern now looks something like this, where we do see radiation of species in this intermediate stage, and then the audacious macrocyclic stages, and of course, lots of bursts of radiation from that sporothallus, just due to biogenic radiation, in addition to the biological specialization that's going along in this axis. Okay, I went a couple minutes over time, so I will just conclude by saying we've still got a lot of hypotheses we are trying to test now in various ways, but we do think that that hetero-macrocyclic life cycle was ancestral to the Rus and actually helps explain a lot of the evolution that we see, at least in terms of species. And I want to thank my funding agencies and all the members of my lab, whether they worked on Rus or not. They were really instrumental in helping my science. So thank you very much.
Thank you very much, Cathie, for an excellent presentation. We are out of time, and I would encourage anyone to send your questions to Cathie via the Q&A or the chat. And at this time, I'd like to introduce